Welcome to the deep dive. Today we are taking a massive shortcut right into what you could call the core engineering challenge of all multicellular life.
It's a fundamental question, really. How do cells organize themselves into these perfect functioning tissues?
Exactly. And how do they know with just absolute certainty when to start growing and, maybe more importantly, when to stop?
It sounds so basic, but that's the difference between a stable, healthy organ and, a tumor. The concept is called tissue homeostasis.
Right. And we've known for a long time, for decades really, that mechanical forces, the simple pressure and crowding from neighboring cells are a critical trigger for these decisions.
A cell can literally sense that it's packed in too tightly and it shuts down proliferation.
But here's the thing. If you talk to cell biologists, the actual connection between that brute physical squeeze and the complex internal biochemical signals has remained, as our sources put it, largely mysterious.
Yeah, the input was clear physical force, and the output was clear. A molecular switch gets flipped.
Yeah.
But the wires connecting them,
totally invisible.
And the revelation we're unpacking today is that those wires, they aren't molecular ropes at all. They're electrical. The resting membrane potential, which we call U, that basic voltage across the cell membrane, that acts as the crucial, instantaneous physical link. It connects mechanical pressure directly to the cell's growth controls.
So we're going to trace a direct physical chain of events for you. It starts with mechanical force changing a cell's internal density. Just how much stuff is packed inside.
And that change in density immediately and physically shifts the voltage across the membrane.
And that voltage is what flips the growth switch. Our mission today is to really understand this elegant physical coupling. Let's dive in.
Absolutely.
So to really understand this, we have to start by challenging a very old assumption in cell biology.
A really foundational one. For years, scientists just sort of treated the density of the cellular contents, the ratio of its mass to its volume, as a constant.
So if a cell got squeezed, everyone just assumed its internal concentration of proteins and lipids, stayed proportional to its size.
And that assumption, according to the sources we're looking at, is just fundamentally incorrect. Cellular biomass density, or RUF, which is the absolute concentration of all the big molecules inside proteins, RNA, lipids.
All the guts of the cell.
Exactly. It's a variable.
Yeah.
And it's a central physiological variable tied directly to mechanical pressure.
Okay, so how did they measure this? I mean, you can't just put a cell on a scale.
Right. This required some really sophisticated tools that could measure concentration without breaking the cell open. They used things like normalized Raman imaging, NOR-I, and holotomographic microscopy. A device called a tomocube.
So these aren't just looking at the shape of the cell?
Not at all. They're quantifying the absolute protein and lipid concentrations inside the cell volume in real time.
And what did they find?
They found a really striking correlation across multiple standard cell lines, MDCK, MCF10A, even human primary mammary cells. As the tissue got more crowded, as the cell number density went up, the cellular biomass density went through a significant observable increase. You are literally concentrating the cell's insides when you squeeze it.
Wow. Okay, so let's bring in the electrical part now, the membrane potential, U.
Right, so U is just the voltage across the membrane. It's usually negative on the inside.
And we can think of this in terms of two key commands. Right?
Exactly. If the inside of the cell becomes less negative, we call that depolarization. And historically, you can think of depolarization as the go signal, grow, proliferate, activate.
And the opposite?
The opposite is hyperpolarization. That's when the cell becomes more negative inside. And that usually signals stop quiescence, differentiation, just arrest.
So we have crowding, which leads to a high concentration, a high growth. What happens to the voltage to U?
It immediately hyperpolarizes. As that biomass density went up because of the crowding, the NDCK cells became more and more negative inside. And this was a universal finding across all the cell types they tested.
So the mechanical state of being crowded instantly generates the electrical stop signal.
That's it. Think about it. The cell is using electricity to signal its own housing shortage.
That is fascinating. And you mentioned this explains some older mysteries.
It does. Researchers studying CHO cells, this was back in the 1970s, they saw that cell density somehow correlated with electrical changes, but they had absolutely no physical mechanism to explain it. This new research provides that missing link.
Okay, but how do we know this isn't just a coincidence? How did they prove that the compression is actually the cause?
That's the key question. They artificially squeezed the cells in the lab. They applied external osmotic pressure using simple things like sucrose or PEG.
So they're squeezing the cells without them actually touching each other.
Precisely. And this forced an artificial increase in biomass density. The result was instantaneous and definitive. The membrane potential immediately hyperpolarized. Wow. It's very strong support for the idea that the change in voltage is a direct, rapid physical consequence of changing the cell's density.
So if we just pause here for a moment, let's do a quick recap. The picture is clear. Crowding leads to high cellular biomass density, and that high forces the membrane to hyperpolarize to create that electrical stop signal. This correlation seems to be the first crucial step.
Exactly. It's a conserved physical response.
All right, now we get to the core of the breakthrough, I think. The physics of how a physical squeeze actually generates an electrical signal, this mechano-electro-osmotic model. How does a higher concentration of proteins actually change a voltage?
The mechanism is just. It's elegant, and it's rooted in very basic chemistry. Inside the cell, the vast majority of those big macromolecules, proteins, DNA, RNA, they aren't neutral. They carry a pretty significant net negative electric charge at physiological pH.
So the cytoplasm is basically a dense, negatively charged gel.
That's a perfect way to think about it.
Yeah.
Now, when an external force compresses the cell has to resist that. It pushes back by increasing its internal osmotic pressure. And that osmotic pressure isn't just driven by the concentrated macromolecules themselves. It's also driven, crucially, by their counterions.
Wait, what are counterions?
Good question. They're the positive ions, like potassium, K plus air, and sodium, Na plus air, that are chemically drawn to balance out that massive net negative charge on all the proteins and nucleic acids.
I see. So for every negative charge on a protein, there's a little positive ion floating nearby to keep things neutral.
Exactly. So when you increase the concentration of the negative macromolecules by squeezing the cell, you also have to increase the concentration of these positive counterions.
Okay, I'm with you. So the tighter the squeeze, the higher the biomass density, and the higher the concentration of these positive counterions inside the cell. Now what happens?
Now you have a massively steep ion concentration gradient across the cell membrane, much steeper than normal. And if you think of the cell membrane as a slightly leaky dam, that super high concentration inside promotes a massive diffusive efflux, a net leak of positively charged ions out of the cell.
So positive ions are just flooding out faster than the cell's pumps can bring them back in just because the concentration difference is so huge.
That is precisely it. And when you have a net loss of positive charge from the cytoplasm, what happens to the inside of the cell?
It becomes more negative.
It becomes more negative. And that buildup of net negative charge is the definition of hyperpolarization of the membrane potential, U. An intrinsic physical response.
That's a really powerful concept. It means the cell doesn't need some dedicated special protein sensor to detect pressure. The physics of this charged material being compressed is the sensor.
And it provides a global, instantaneous readout that bypasses a lot of other confusing factors.
Like what? Why is this better than other ways a cell might sense being squished?
Well, think about it. Cell shape and size can change drastically in a dynamic tissue. A migrating cell looks totally different from a resting cell. But this density readout is independent of changes in cell size or its surface to volume ratio. It's a clean, reliable measure of internal concentration.
And to really prove that charge was the key, they didn't just look at density. They actually went in and manipulated the charge of the biomass itself.
They did. This is a brilliant experiment. They engineered cells to express either a highly positively supercharged GFP or highly negatively supercharged GFP and grew them next to normal cells.
Okay, and what was the prediction?
The model predicted that if charge really mediates this effect, then increasing the internal positive charge should actually make the cell denser because it would attract even more negative counterions and increase its own osmotic pressure.
And did it work?
It worked perfectly. The positively supercharged cells were demonstrably denser than their control neighbors, and the negatively supercharged cells were more dilute. It confirmed the physics.
Wow.
Yeah, and they even blocked protein degradation with the drug, MG132, which caused internal density to build up. And just as predicted, they saw instant hyperpolarization. The mechanism is airtight.
So let's recap again. Mechanical pressure is translated through the physics of a charged biomass density into a reliable electrical voltage user. So now the big question is, how does this electrical signal actually switch the cell's fate?
Right, so now we move from the physics problem to the biology problem. How is this executed? The experiments confirm that this voltage, U, is the primary regulator of biomass production and proliferation.
So they could override the cell's natural density sensors just by messing with the voltage.
Completely override them. Let's look at the growth signal first. Artificial depolarization. If you make a cell artificially less negative inside, it should interpret that as the go signal, right? It should think, I'm not crowded, I need to grow more.
Okay, so how do they do that?
They used a few methods. Drugs like uvein or digoxin, which block the sodium-potassium pump, making the cell interior less negative. They even used the bacterial sodium channel to do the same thing.
And the result?
In every case, the cells completely ignored their normal density limit. They just kept proliferating until they reached a significantly higher biomass and cell number density. The depolarization signal drove this unchecked growth.
That is a huge finding. A simple electrical change makes the cell ignore its physical crowding and just overgrow. What about the stop signal?
They did the reverse. They induced hyperpolarization, making the cell more negative, using a drug called valinomycin. This forced the stop signal.
And what happened then?
Even when the cells were sparse, with plenty of room, they prematurely stopped growing. They plateaued at a much lower non-physiological density. It confirms that U is the master control loop for tissue density.
So we have the electrical switch. Where does that switch connect to the molecular machinery? The sources point straight to the famous Hippo pathway.
Yes, the Hippo pathway is the classic system for sensing mechanical force. It controls cell proliferation through a critical molecule called YAP.
Yes, associated protein. I remember this. The rule is simple, right?
Very simple. YAP in the nucleus means go. It promotes growth. YAP trapped in the cytoplasm means stop. It suppresses growth.
And U directly controls where YAP goes.
Absolutely. The research showed that in sparse, uncrowded cells, if you artificially induce hyperpolarization, the stop signal YAP gets kicked out of the nucleus and trapped in the cytoplasm. Growth stops.
And the reverse. In a crowded dish of cells.
In dense, crowded cells, if you artificially depolarize them, give them the go signal, YAP re-enters the nucleus, overriding the physical compression and promoting growth.
So U is clearly the upstream command. It's not about the nucleus being squished or something simple like that.
Exactly. Which brings us to the final molecular player, a transmembrane protein called FAT1. It's known to be an upstream part of the Hippo pathway.
And this is the final link in the chain.
It is. The sources establish that U controls the assembly of the Hippo signaling complex, specifically a protein called MST1, right on FAT1's tail inside the cell.
How does that work physically?
When the cell is hyperpolarized, that stop signal voltage causes MST1 to form these organized clusters on the FAT1 tail at the membrane. This mimics the Hippo-on state, which ultimately keeps YAP out of the nucleus.
So if you get rid of FAT1...
If they completely removed FAT1 from the cells, none of the voltage manipulation had any effect on YAP anymore. It proves that U activates the FAT1 Hippo axis in response to pressure.
That's a complete picture. Physical pressure, sensed electrically, with that electrical signal dictating the activity of the core mechanical growth pathway. Amazing.
And this deep dive into the physics of homeostasis immediately clarifies dynamic processes, things like wound healing. The cell doesn't wait for slow biochemical signals to diffuse around. It uses electricity to coordinate repair instantly.
Tell us about that wound healing case study. What did they see?
They took a consolate sheet of cells, a monolayer, and just made a scratch wound in it. As the cells at the edge start migrating to fill that void, a wave of mechanical stretch propagates into the tissue.
So the cells are being pulled and relaxed.
Right. And this stretch causes a concurrent drop in their biomass density in rho. And critically, a corresponding wave of depolarization, the go signal, starts at the wound edge and moves inward.
So the electrical signal, the hey, you're uncrowded, time to move signal, is sent out ahead of the physical movement.
Exactly. This depolarization wave actually precedes the reorganization of the cell's internal skeleton and enhances healing because it signals distant cells to start getting ready to move and divide before they even feel the empty space.
And this isn't just some complicated system in mammalian cells, is it? The sources suggest this is ancient.
It is fundamentally conserved. They observed the exact same response, this electrical coupling of mechanical force in regenerating frog embryos after tail amputation, and even in the simple freshwater polyp, Hydra.
Wow.
When you see a physical mechanism like this conserved across all those different branches of life, it tells you how reliable and essential it is.
The implication for future research seems huge. I mean, if U is this reliable, size-independent electrical readout, it could modulate any signaling pathway that starts at the plasma membrane, not just Hippo.
That's a great point. And the sources actually confirm they saw effects on the MAPK pathway too, which suggests this is a much more universal physiological sensor than we realized.
And the coupling works both ways, right? Pressure affects you, but changes in you can affect pressure.
Exactly. It's a complete feedback loop defining the tissue architecture. If a cell suddenly misexpresses an ion channel and changes its U, it will change how it perceives its environment and how much pressure it exerts on its neighbors.
So we've established that elementary physics, this beautiful coupling of charged biomass density and membrane potential, provides cells with the essential instantaneous information they need to manage growth and coordinate tissue behavior.
And this brings us full circle, really, back to the clinical relevance. The sources highlight something we've known for a while but couldn't explain. A very common phenotype in many tumors is a depolarized resting membrane potential.
And we now know that in this density sensing system, depolarization is the explicit electrical signal that tells the cell, keep growing, ignore your crowded state.
It's the go signal stuck in the on position.
Which raises a really crucial and I think provocative question for you to consider. If depolarization is the keep growing signal that drives this uncontrolled proliferation, how might future cancer therapies move beyond complex genetics and exploit the fundamental physics of the cell to actively restore that hyperpolarized stop growing signal?